Assalamualaikum warahmatullahi wabarakatuh. Today we are from Group 5. We will present about the advantage and disadvantage of plant tissue culture. First of all, I would like to introduce my teammates. Myself, Muhammad Ali Badrul Majid. My metric number is 2022-622-064. And then we have Nur Aisyah binti Abdul Halim. Her metric number is 2022-481-8928. And then we have Aina Dania binti Zulhusni. Her metric number is 2022-890-902. And last one, we have Shahira Nufiti binti Saiful Faizal. Her metric number is 2022-478-824. Next, I will explain about the definition of tissue culture. Tissue culture is the fundamental science influenced by biology. It involves the cultivation of desired cell, tissue, or organ in a sterile synthetic medium. The conditions such as temperature, light, and humidity are controlled during the process. Micropropagation, also known as the plant tissue culture, this process allows for the rapid and uniform growth of multiple plants. It provides a controlled environment for the development of plants at the cellular level. Next, the top perspective also highlight the importance of control condition in the growth of plant, cell, tissue, and organ. In summary, plant tissue culture play a vital role for advancing our understanding of plant biotechnology, also help the valuable compound in the commercial sector. That's from me. I will pass to the next one.
Next, the advantage of plant tissue culture is we only need small required amount of initial plant. This is because we use a small amount of parent plant to make a lot of new plant with the media. We can only use the small amount and put it in 1 cup of media, and we can make it a lot in one time. So we didn't have to use soil and have a big space. Next, the new plants are more likely to be free of viruses and diseases. This is because tissue culture helps us to produce more copies of same plants with desired character like big fruit, more fruit, colorful flower, dead and disease resistant. This is because we can control the temperature and the environmental to make sure the plant or our tissue culture product will not contaminate. So we can always make sure and we control the growth of our tissue culture plant. And
Next, the advantages of plant tissue culture is the process is not dependent on the seasons and can be done throughout the year. This is because tissue culture helps us to produce plants which do not depend on the seasonal changes of the area for a particular year. This is because seedlings are made available, planting is possible all throughout the year. Lastly, plant tissue culture just needs a small space. This is because in tissue culture techniques, very little space is needed because in this technique, we culture only a small part of tissue in the nutrient media containing fluids, so a very small space is required for its maintenance.
Last presenter is me. My name is Aisha and I'm going to talk about disadvantage of plant tissue culture. First of all, it costs more money in building the facility. We already know that tissue culture may require more labor, and also in order to equipping the lab with the equipment and also the chemical, we need to use money. Second, it have chance that the propagate plants will be less resilient to disease. This is because when we try to grow the plant outside of the lab, the plant may face difficulties to adapt with the environment outside of the lab. Lastly, it's easy to be contaminated by living organisms such as bacteria and also yeast. That's all from our groups.